Hello everybody. I am Max Tassell. I'm a PhD student based at Cardiff University, and I'm going to talk today about invertebrate sampling. So this is the collection of animals without a backbone, which is the vast majority of the diversity of life on Earth. And this is for, ironically perhaps, National Hedgehog Awareness Week and the Hedgehog Friendly Campus at Cardiff University, which is an important endeavour to make our campus as friendly as possible to hedgehogs so that they can exist happily on the campus. So I'm going to speak a little bit about the history of invertebrate sampling, a kind of overview of why it's important, and I'm going to finish off with just a few things you can do at home to sample invertebrates, which are easy and quite simple and reliable methods that you can use yourself. It's a bit of a whirlwind tour. You can you can do many lectures and talks on invertebrate sampling. It's a very broad topic as a result of the diversity of invertebrates themselves. I'll go into this a bit, but a lot of different invertebrates need different sampling, need sampling in different ways if you want to collect them. So without further ado, let's get into the talk. So invertebrate sampling has a very long history. You could say that humans have been sampling invertebrates for as long as we've existed. So people have actually been collecting invertebrates as for food, for medicinal reasons, as poison to tip their arrows or spears, as well as for recreational reasons like psychotropic effects, and that feeds into more spiritual reasons. During rituals and things like that. Over 100,000 years or so of our history, insects are fundamentally vital to human evolution and human survival. But if we want to go very much towards the present, Aristotle collected a lot of invertebrates during his lifetime. This was in the 4th century BCE. Aristotle was Plato's student in ancient Greece, and he described about 500 types of invertebrates. It wasn't quite species or anything like that. It was his own way of kind of categorizing animals, and he described about 500. If we then jump further on to Linnaeus and Fabricius, they formed many of their ideas about taxonomy generally by looking at plants and invertebrates. Linnaeus wrote Centuria Insectorum, which describes about 100 species of insect and crustacean. Crustaceans were previously considered insects. You might think that's a bit silly, and so do I. But there we go. Crabs were considered insects back in the time of Linnaeus. But for Fabricius, his student, was far more productive when it came to describing invertebrates, and he described nearly 10,000 species. So he absolutely wiped the floor with his mentor Linnaeus, and he really set the gold standard for insect description. Charles Darwin and Alfred Russel Wallace had a keen interest in entomology. And Charles Darwin was a member of the Royal Entomological Society, then the Royal Entomological Society of London. And this here is the sign-in book in Mansion House in St Albans, which is where the Royal Entomological Society headquarters are based. And I had the privilege of, during my master's degree, of visiting and looking in the sign-in book. And this is a photo I took. And you can see here, this is Charles Darwin's sign-in signature. So he's had a long history of entomological science and thought, and many of the theories and ideas in evolution and ecology have been evidenced using invertebrate sampling. So the theory of island biogeography is an extremely important theory in ecology. How many species exist on Earth? the ideas of coevolution, where two species are kind of evolving together, food webs as a concept, ecosystem health, bioindicators. These are just a few examples that were evidenced using invertebrate sampling. So it's really, really important. So why is invertebrate sampling useful and important? So the vast majority of species are actually invertebrate species, to our knowledge. And when I say to our knowledge, I mean we know that is categorically true, but what we call invertebrates, so arthropods, mollusks, nematodes, etc., they are probably the most diverse group of animals on Earth, but there could be other groups such as bacteria or fungi or protists, archaea, that may be more diverse, but the whole concept of a species becomes quite muddied in these groups. So I won't go into that argument. But from what we know now, the vast majority of species are what we classically consider invertebrates. So the arthropods, the mollusks, the annelids, etc. So vertebrates are cool. They are incredibly fluffy and cute, and currently 66,000 species of vertebrates have been described, but this is less than 5% of all of the described species. So of course hedgehogs are great, but the vast majority of described species are actually invertebrates, so 1.3 million extant vertebrates, so that is living vertebrates, have been described. Most of the remaining undescribed species are also invertebrates. So the vast majority of species on Earth are actually invertebrates. So they're obviously very important. And you see here on the right, this is the biomass distribution on Earth. So you can see here in A, you have plants. They're a huge proportion of biomass, so that's 450 gigatons of carbon. So this is how biomass is typically measured. You see animals are only 2 gigatons of carbon, and half of that are arthropods, so that's the insects, the crustaceans, the centipedes, millipedes, etc. So half of all animal weight on Earth is made up of arthropods alone. So that doesn't even include other invertebrates like mollusks. You can see 0.2 gigatons of carbon. Humans, by comparison, are 0.06 gigatons of carbon. So that's 6% of all of the arthropods on Earth. So you can see that basically animals make up quite a small proportion of the total weight of life on Earth. But of that proportion, arthropods are about half. So obviously animals being a very important aspect of the diversity of life and the biomass of life, arthropods alone are half of that. Vertebrates, so your hedgehogs, your tigers, your humans, are an extremely low number, extremely low weight total amount. So figuring out how to collect and identify this enormous group is essential for many large-scale ecological analyses or surveys, like if you want to determine whether a certain area is suitable for development or is suitable for hedgehogs, for example. Sampling the invertebrates is a really important step you could take. And just following on from this, maybe this could be a conceptual framework for conservation. This is taken from Insect Conservation: A Global Synthesis by Samways, which was published last year. So you see like the scale of the beetle, the elephant, and the tree are determined by the relative contribution to global biodiversity measurements in the groups that they represent. So elephants here represent vertebrates, beetles here represent invertebrates, and the tree represents plants. So you can see in the left-hand figure, the beetle vastly outsizes the other two groups because invertebrates are by far and away the most diverse group on Earth. So that's compositional biodiversity. Then in terms of biomass, we see a slight change. We see that beetles the beetle is larger than the elephant, but the tree is vastly larger than both. And that's in terms of biomass. So we saw that in the last image. So we see plants are just overwhelmingly much more important in terms of total biomass to the life on Earth. So that's structural biodiversity. And then you have the number of interactions between species, which is functional biodiversity. And you see that, again, invertebrates are much larger than vertebrates. And then again, plants are larger than invertebrates again. So maybe we should really judge the importance, and maybe we could kind of direct conservation based on these ideas. Instead of what's fluffy and cute like hedgehogs. I realise I'm being highly ironic here speaking for this event, and we will get on to hedgehogs because they are important, of course. But as US President Joe Biden said as a young man in 1727, surveying only vertebrates is viewing an important but tiny section of the diversity of life. I may be paraphrasing, it may not even be a quote from him, I'll let you decide. We should aim to sample as wide a variety of life as we feasibly can. So that obviously includes vertebrates and invertebrates, plants, as much as possible, but sometimes that isn't feasible. But observing patterns of diversity in different areas can teach us many things, from ecosystem health to species interactions and evolution, and the full diversity of life is the best possible way you can look at that. And this has application to industry and environmental management. It's really important to determine how feasible a specific sampling regime can be. Even just sampling all of the invertebrates in a given area may be too difficult. But some invertebrates should be surveyed for many ecological analyses. So what about the hogs? Yes, of course, this is about the hogs to some degree. So sampling for invertebrates can directly help us examine the suitability of areas for many other species, including hedgehogs. So hedgehogs love consuming invertebrates. They eat lots of beetles and caterpillars, earthworms, slugs and snails, etc. So surveying for these groups can actually help us determine whether an area is suitable for hedgehogs. And this extends to many other terrestrial species that rely on insects. So you can, and that is most terrestrial species. So most terrestrial vertebrates, to some degree, even indirectly, rely on invertebrates. So invertebrates may pollinate the trees that they eat, for example, even if they don't directly interact with the insects. So we can aim to sample groups that are likely to be important for another species of interest, in this case the hedgehogs. So you can sample for beetles or caterpillars, for example, because they are very important sources of food for them. So what does invertebrate sampling look like in research? So this might not be applicable to you, but this is the sort of the standardized approach for invertebrate sampling, if such a thing exists. A typical pipeline for invertebrate sampling. So this is kind of based on what I've been taught and what I did for my PhD sampling. So you kind of determine your collection methods, and that's based on what you want to find. Different. We'll get into this in a bit, but different collection methods can collect different types of invertebrate, basically. So you'll kind of determine what you want to collect, and from that, that will give you what sort of collection methods you might want to use. Then you formulate a sampling design. Do you want it to be systematic or random along a transect within quadrats, for example? And then after you've determined this, you can deploy your traps or use your sampling technique. Then you can transfer your invertebrates into collection tubes, bags, etc. And do you need to keep those invertebrates alive? Like you might want to observe them for behavioral studies or experiments. If you don't need to keep them alive, you will probably need to kill them to identify certain groups. That's not true of many groups. You can certainly identify them whilst they're alive. Some you might need to kill them, unfortunately. And then you can sort the samples and identify those specimens to whatever taxonomic rank of interest you desire. So that might be species, genus, family, order, etc. And that can often take a very long time if you're going to species level. So this is what I did for my invertebrate sampling regime. So this is Round Island. This is my field site. I won't get too much into my PhD because this is just an example of what you might do. So you might generate a map of your sampling locations. So this was actually randomly generated. And then you'll place a pitfall trap in the center of the quadrat, and you might collect it after 48 hours. So I decided to go with pitfall traps because they collect the groups of organisms I want, and I left them out for 48 hours because I wanted a sort of like long collection time to get a good amount of invertebrates. But as well as that, I used a leaf blower because, and this was modified into a suction sampler, so it didn't actually blow, it very much sucked. And these two collection methods collect different groups of invertebrates. So the pitfall traps don't collect the same invertebrates as the leaf blower, broadly speaking. So we're actually getting a sort of greater diversity, a sort of a better total picture of what's present in these quadrats. I also then implemented hand collection, specifically for ants, because I realized that many of them would be sort of in rocks, in rotting wood, etc., etc., and the other methods wouldn't necessarily catch them at the efficiency level that I required. So that's kind of what it looks like. You determine where you want to sample, you then set your traps out or employ your method of collection, and then you would later take them back to your house or the lab and you would identify them. So that's what it looks like in research. And there are just many different things you want to think about in research. So all of these different areas in the different squares are actually sort of, you would have to use a different sampling method to sample invertebrates in those areas. So on the tree trunk, you might choose hand sampling. You might be looking under the bark, and you might be using something called a puta, which you use to suck up. Quite comically named, I always feel, to suck up the invertebrates that you're finding. And you might try to standardize it, so you might want to be doing hand collecting in 15-minute bursts on a single tree trunk, and then you'll move on. So that's just sort of one example of what you might use on a tree trunk. But if you then look on the ground, you might want to use pitfall traps. If you look in the canopy, you might want to use different methods like like foliage beating or foliage extraction, where you actually cut off a bit of the tree, you bag it up, and then you take it back to the lab and extract all of the arthropods or invertebrates generally from those samples. So there are lots of different sampling techniques for different invertebrate groups. But then also the actual habitat and the structure of the habitat can determine what sampling technique you'll also use. So different techniques will collect different invertebrate groups. You might use nets, pots, lures, hand collecting, visual surveys, etc. And it all depends on what you're trying to find and the area in which you're working. So this is a diagram showing different sampling techniques and the efficacy with which they catch different groups. So if we go to dragonflies, for example, fourth from the right, we see that active visual surveys are a really efficacious method to sample for them. So that would involve, say, walking on a transect and counting the number of dragonflies and the species of dragonflies you see along a specific route in a given amount of time. And we won't go into all of these because that would take far too long, and you may find it far too boring. But this is the sort of useful information that entomologists use to determine what they want to sample and what sampling methods they will use to sample those groups. There are lots of different methods because there are lots of different invertebrates, basically. Let's start a bit slow. Let's start some simple but effective techniques you can actually do at home. So we're just going to give a quick overview of pitfall trapping, hand collecting, and visual surveys. So this is a pitfall trap. It essentially is a cup in the ground that invertebrates will walk into and not be able to escape from. You can see here pictured is a beetle, I think, in this cartoony form. And these are really, really simple to set up. They're really, really effective, and they're very, very cheap. They are a standard collection technique that every entomologist should and pretty much does know how to use. And they're a really, really great sampling technique. So these capture ground-dwelling active invertebrates like beetles, ants, centipedes, things like that. Typically, the active invertebrate will be walking along the ground and then will tumble into the cup or container. And these primarily measure activity density. So sort of the greater the level of activity in a given area, the more invertebrates you'll find, essentially. They're simple, they're cheap, they're effective, and they're reliable. You can also add bait to select different groups. Typically, you would also add some liquid in the bottom of the pitfall trap to prevent escape and to preserve the different invertebrates and to prevent them from eating one another. But you can also run them dry, so you have no liquid in the bottom and it doesn't kill them. The only thing you need to know about that is that if you are running them dry, if there's no liquid in the bottom, you can't leave them out for too long. So I'd say that if you do want to run them dry and don't want to kill the invertebrates, then anything less than a day I think would be fine. But anything longer than that, you might need to add some liquid. So you can really do this with any sort of cup or container that you're happy putting in a muddy hole in the ground. You'll need a trowel, a shovel, or your mitts to dig a hole. You dig the hole big enough to fit the cup and put the soil aside in a pile. The trap goes in the hole, of course, and then the soil pile you've made is used to make sure the lip of the cup is flush with the ground surface. It's really important that the lip of the cup is flush with the ground surface so that the invertebrate essentially just falls straight in. It doesn't have to go up. It can't wheedle in between the lip of the cup and the soil. It just sort of is going about its business, and then whack, it is trapped. So that's how you use them. They're very simple. You can find, as with all of the three monitoring techniques we're looking through, you can find how-to videos on YouTube, for example, to see a sort of practical guide. And I may be running a few practical sessions later on, which we might be advertising later. So that was pitfall trapping. This is hand collecting. So this is actually a pooter, or as the Americans call them, an aspirator. And this is used to suck up invertebrates. They can be quite simply made at home as well. So this is a jar, and two straws have been attached to the lid. As you can see, one of the straws is covered by sort of a gauze or a piece of cloth, so that when you suck in the invertebrates, they don't then get sucked into your poor lungs, and the invertebrates basically stay in this jar. Pooches are excellent because they allow you to observe the invertebrate that you sucked up, because obviously you have it in a jar then, and it's contained. So you can often you can suck up an invertebrate and then you can observe it, and you can even identify it a lot of the time and then release it. But as well as pooters, you can use a whole range of different additional tools during hand collection. So you can use sort of fine tweezers or paintbrushes, small paintbrushes, like a magnifying glass to identify things, or even just collection tubes. I feel that's really all you need, actually. Collection tubes is often the only actual piece of equipment you need during hand collection. You can simply scoop things up into collection tubes. This is perhaps the most varied collection method, but it often collects the highest diversity. It's harder to standardize. So pitfall traps are very easily standardized because you can account for the volume of the pitfall trap, the time they're left out. Different people setting pitfall traps are likely to be doing it in a fairly similar way. Whereas hand collection is harder to standardize because searching haphazardly in a given area will vary between people. So some people might be quite skilled at collecting certain groups, whereas other people might have a tougher time with one group and more easily sample another group. And also the effort might vary. So some people might basically be really great at searching tiny things. Like they might be a bit of a perfectionist when they're sampling. Other people might sample the quadrat sort of more wholly, but a bit less perfectly. So it's a bit harder to standardize. It's very good where in other habitats where other methods don't work. So e.g. Under rocks or in rotting logs, very structurally heterogeneous habitats, or for species where other sampling doesn't work, or you only want to collect sort of one type of invertebrate or a small group of species, that sort of thing. So as I say, all you really need are collection tubes. A pooter takes your ability to capture small invertebrates to the next level, so you can suck up specimens and transfer them to collection tubes. As I say, you can use other tools, tweezers, a small brush, hand axes, and hammers can be quite good for breaking open stones or wood, that sort of thing. And they can be useful, but of course you don't want to, unless you're using these invertebrates for really quite important research or studies. You don't want to be breaking open rocks and destroying rotting logs because they're really important sources of resources for invertebrates. But they can be useful if used wisely. And this is a very immediate method. So you can collect your invertebrate, you can observe it, and then you can preserve it pretty much immediately after collection. So it's very immediate, though the next method we're going to go over is also fairly immediate. Whereas pitfall traps, you need to wait a whole day, maybe 2 days, until you actually collect your sample, for example. So the final one we're going over today is visual surveys. So this is a really fun one. You're basically watching or observing invertebrates at given resources, for example. And it can actually be quite difficult, so it kind of feels a bit like this sometimes, because invertebrates are quite fast. They can fly around. You might be trying to identify butterflies or dragonflies on the wing, and that can be really difficult sometimes. So it's not just standing around. You might be physically standing around, of course, but it's actually quite hard work sometimes. So you need to keep account of what you're seeing. It can require quite good identification skills for a specific group. So identifying solitary bees on the wing, for example, is really difficult for a lot of groups, especially the smaller ones. Hoverflies and dragonflies also really difficult to identify sometimes on the wing, depending on the species, of course. And visual surveys usually involves watching specific resources. So flowers, for example, or walking a transect of a given length in a given time and recording what species and how many you see. So in practice, all you need is a way of recording what you see and timing your session. So you might want to sit in the park and count the number of butterflies you see, but you can go a step further by recording what species of butterfly you see. Or you can do the same thing with bees or hoverflies on flowers or dragonflies along a walk along the canal. It can be again difficult to make comparisons between recorders because of different skill levels or different abilities to see the invertebrates, but can be a reliable measure of activity if you do some mathematical shenanigans. So using multiple sampling methods reveals a wider range of invertebrates than relying on just one technique. But as we've seen from those three methods, sampling methods can be simple, cheap, and reliable. You can do pitfall trapping for a pittance. You only need some collection tubes to do hand sampling, and all you really need is a notebook and some time to do visual surveys. So you can actually do all of these really cheaply. You probably already have a lot of the tools required to do these. Identification to species level can be tricky in some groups, but is really rewarding to learn. I mean, if you're really interested in bees, for example, Stephen Falk has amazing Flickr resources, and he's, of course, I would strongly encourage you to buy his bee identification manuals. If you're looking at butterflies or moths, again, there's a lot of really great resources that you can use for UK species, and they're some of the groups that a lot of people start with. So I would start with any groups you find exciting, and then next thing you'll know, you'll be an entomologist. You'll be looking at all of the tiny things. You'll be looking at beetles in the soil. You'll be looking at ants. You'll be looking at sort of non-hoverfly flies. You'll be looking at a whole range of cool things, not to mention aphids, of course, which are great. Hedgehogs love eating beetles and caterpillars, so if you want to see if your garden or a local area could support hedgehogs, you might want to try pitfall trapping with permission, of course, for beetles, and you might want to do some hand collecting for caterpillars. You obviously don't necessarily need permission to do hand collecting in public spaces. So that's a way you can see if hedgehogs could potentially survive in a given area, if an area is suitable for them. Many trapping methods, as I said, can be used without killing the insects. So again, you can try dry pitfall trapping if you're setting the pitfall for less than 24 hours, and to make sure to place anything caught by hand gently back to where they were found. As for many groups, you don't need to kill them, to identify them, that is. There are many other excellent resources to learn about insects, surveys, different sampling techniques that require more specialist equipment. The best place is probably YouTube. There are a lot of other resources as well. But YouTube's a really great place to start because you really get that practical how-to videos that are great for more complex sampling types. If you really get into your insects, this talk has been such a whirlwind tour. I can only apologize for how rapid it's been, but there's so much to talk about. You really can't cover a lot in a short space of time. Very importantly, the Royal Entomological Society has 1 year free membership for students, which is possibly the best deal in history ever. for students, because the Royal Entomological Society, as we heard, Charles Darwin was a member. It is one of the best organizations you could ever join if you're interested in invertebrates. They don't just do insects, even though they're called entomological. They do spiders. They even do slugs and snails to some degree. So it's really the Royal Entomology and Allied Animals Society. And as I say, yeah, it has 1 year free membership for students. So go ahead and join. You will not be disappointed. It's a really great community. And there's some really exciting things happening with the society. If you are interested, just go to the Royal Entomological Society website, which you can just type into Google, the Royal Entomological Society, or you can send me an email or hit me up on Twitter or anything. Yeah, but basically join if you're interested. So just a few take-home messages. Invertebrates are very important. Surveying for them can be very simple, but also very fun. Monitoring invertebrates is very valuable because they're so important, but we need far more people doing it. Invertebrates, such as hedgehogs, are great and can provide important sources of funding for invertebrate conservation as umbrella species. So this really means that by preserving hedgehogs, we can try to preserve the ecosystems on which they rely, and that includes the invertebrates. But ultimately, we need more dedicated resources and long-term monitoring schemes, which all originates, of course, from invertebrate sampling. So hopefully this has given you a bit of a whirlwind tour of the world of invertebrate sampling, and it's given you 3 different methods you might want to have a try at home to see if you can find some cool invertebrates. And if you want to check out if your garden is suitable for hedgehogs, then you can sample for some beetles and some caterpillars using pitfall trapping and hand collecting. If anyone has any questions, of course, feel free to email me or get me on Twitter at MaximumInsect. If you haven't guessed, I really like insects. So I hope you enjoyed this incredibly disjointed and chaotic talk, and I hope you have a wonderful day, and I hope you go and sample some insects and have some fun and become a hedgehog-loving entomologist, basically. I hope you become an entomologist. Cool. Bye-bye.